Welcome to the National Center for Complementary and Integrative Health, or NCCAH Hot Topic webinar on probing interoceptive processes at behavioral, psychological, and neurophysiological levels. I'm Wen Chen, a branch chief and program director at NCCAH. I'll be serving as the moderator and as a discussant at today's webinar. Before we get started, I would like to share today's webinar agenda, introduce our speakers, panelists, and share some house or internet keeping issues. We will begin the webinar with an overview by Dr. Ellen Langevin, Director of NCCAH. Dr. Langevin will be followed by 3 speakers. Due to a unexpected technical issue, we will change the speaker order slightly today. So the first speaker will be Dr. Eleni Frangos from the NCCAH Intramural Program, presenting Assessing Interoceptive Neural Networks via Vagus Nerve. The second speaker will be Dr. Ginny Park from Emory University School of Medicine, sharing with us her work on micro-neurography technique applications in interoceptive research. And the third speaker will be Dr. Wolf Malin from UCSF, speaking about variations of interoceptive experience at the interface of mind and body. During these talks, please feel free to email your questions to nccahwebinarq@mail.nih.gov. The address is right at the bottom of the slide, or use the NIH videocast feedback form. We will try to get to as many of your questions as we can during the Q&A session. Following the 3 speakers, our panel discussants representing some of our sister NIH institutes and offices will share with us their perspectives related to today's webinar topic. Each discussant will present a slide, speak for about a minute or 2. We'll begin with Dr. Janine Simons from the National Institute on Aging, NIA. Then Dr. Jim Knott from National Institute on Neurological Disorders and Stroke, NINDS. Dr. Steve Grant from National Institute on Drug Abuse, also called NIDA. Dr. Alexandra Vicentic and Dr. Sivash Vaziri from National Institute of Mental Health and NIMH, as well as Dr. Dana Slosser from the NIH Office of Behavioral and Social Science Research, OBSSR. And we'll end with Dr. Emily Edwards and myself from NCCAH. Again, you're welcome to email your questions to us at nccahwebinarq@mail.nih.gov anytime during the panel discussion. During the panel discussion and webinar Q&A session, the speakers and panelists may also ask each other questions. We hope to end the webinar at 2:30 Eastern Time. If you're not able to watch the entire webinar, please feel free to visit the NIH videocast website later to watch the archived videocast of this event. Now, without further ado, Dr. Langevin, please share your screen to begin the webinar overview.
Well, thank you very much, Wen. It's a great pleasure to be holding this event virtually. I'm going to be, whoops, not this. Sharing my slides. Can you all see the slide? Yeah, good. And so I will be just giving a little bit of an introduction to this webinar, placing the various talks that you'll be hearing in the context of interoception. So why is NCCAH interested in interoception? Well, interoception is a context, right, that has a lot to do with the mind and body interface, right? And NCCAH is interested in practices, treatments that we call mind and body that really also have to do with the mind-body interface. So just to go into a little bit more detail about this, what types of practices are we talking about with treatments? Well, you have some practices like, for example, meditation, where what we call the therapeutic input of the intervention or the practice is primarily mental, but can have effects, physiological effects on the body. And then you have other types of practices, like, for example, manual therapies or acupuncture, where the primary therapeutic input is physical, but then it can also have an effect on the nervous system. And then you have therapies that are sort of mixed, like Tai Chi and yoga, where you have a mixture of mental and physical therapeutic input, and also therapy and effects. So these types of mind and body, both input and output or effects, we believe that the research on interoception is very relevant to these types of therapies. So last year, the NIH Blueprint Initiative sponsored a workshop that was led by NCCIH on interoception. And this was a fascinating workshop where there was a lot of discussion about the definitions of interoception. What does it mean? What does it include? And also, what are the gap areas in research that are needed, both on the basic side and also on the clinical side, to move this field forward, with particular interest in developing research that will then enhance our understanding of mind and body therapies. So what is exactly the definition of interoception? Now, that is not something that is a simple thing, and people are not always in complete perfect agreement. And there was a lot of debates at the workshop about how do you define interoception? And this is the definition that was kind of the consensus that emerged from this particular workshop. Now, there are other definitions, but for this particular, at this particular workshop, this is kind of what we landed on. Processes by which the body senses, interprets, integrates, and regulates signals from within itself. So just to give a little bit more meaning to these words. So what do we mean by within itself? So these are signals that emerge from the interior of the body, the inside of the body, as opposed to signals that emerge from the outside. For example, hearing does not include hearing, taste, smell, sight. It does include input from for example, musculoskeletal tissues, but deep to the skin. And this is where there's some debate as to, for example, proprioception is part of interoception. And so what we decided is that as long as the signals originates from tissues that are deep to the skin, then we would include them as far as interoception. And Dr. Mailing is going to be talking about some of those types of interoceptive signals that are part of that category. Importantly, these processes, the process of sensing, interpreting, integrating, includes both conscious and unconscious phenomena or factors and mechanisms. And the other thing to point out here is that there are different types of interoceptive signals. Some of them are neural and some are extraneural. For example, hormonal signals, right, that derive from the gut, the viscera, that then feed back on the brain via a chemical or humoral or neurohumoral or humoral pathway, are considered extraneural, but they can still be interoceptive. So this particular workshop, we're going to be focusing on neural circuits, and also this was the focus of a recent concept clearance that we presented at our council back in February on functional neural circuits of interoception, and this is what we're going to be talking about today. So what are some of these neural pathways? So we built some diagrams to sort of, and the various speakers are going to be referring to these diagrams because the anatomy of these different neural circuits is complex. It involves a lot of the what we call the autonomic nervous system, right? With different parts of the autonomic nervous system. So I'm just going to describe them very briefly for you right now. So as far as ascending neural pathways, I was hoping to be able to use my little drawing tool here, but I don't see it. So I guess I will point with, hopefully you can see my pointer. So as far as the ascending neural pathways as a part of the autonomic nervous system, you have some pathways that are part of the autonomic nerve, the parasympathetic nervous system, where the afferent pathways arising from, say, the gut or the heart go up the ascending pathways through the nodose or jugular ganglion to the brain via the vagus nerve. Then you also have pathways that emerge from for example, musculoskeletal tissues that will go up via spinal nerves, right? That also the synapse and dorsal ganglion, and then go up to the brain that way. Now, these pathways will then involve and kind of end up in regions of the brains and the synapse in various parts of the brains, including the brainstem. For example, the nucleus of tractus solitarius will also then synapse back up again one other level to areas, for example, of the hypothalamus. And then there are signals that, and we're going to be hearing quite a lot about this today, that that will then sort of generate input to structures of the brain, limbic and paralimbic system, in particular the amygdala, cingulate, prefrontal cortex, areas that have to do with emotion regulation. Now, as far as descending neural pathways, again, you have some pathways that will go down in the autonomic nervous system via either the parasympathetic efferents via the vagus nerve, and then also sympathetic efferents that will also have various different routes, either synapsing through paravertebral or prevertebral ganglia, and innervate various structures such as the heart, blood vessels, gut, etc. So as you can see, these pathways are complex, and just to help people keep reminding people, as we're going to go through the various talks, we're going to point out which part of this anatomy these talks are referred to. So our three talks, so as Dr. Chen mentioned, we're going to change the order a little bit. Dr. Frangos, accessing interoceptive neural circuits via the vagus nerve. Dr. Jeannie Park, micro-neurography techniques, applications in interoceptive research. And Dr. Mailing, variations of interoceptive experience at the interface of mind and body. So which parts of the nervous, the interoceptive pathways do these talks going to address? So first, Dr. Frangos. Oh, here we go. I can annotate. That's what I was going to do. Here you go. So I'm just going to highlight the area that Dr. Frangos is going to address. So we have here the ascending vagus nerve, and Dr. Frangos is going to talk about both afferent and also a little bit about efferent, mostly afferent. And then she's going to be referring to the brain areas that I mentioned, in particular the insula. Dr. Mailing is going to be talking about those somatosensory afferents that I highlighted earlier, that arises from the musculoskeletal tissues and goes up via the spinal nerve. So I'm going to hand it over to, first is going to be Dr. Who's going to be first? Dr. Frangos?
Yep.
Yeah. Okay, good.
Hi, everyone, and thank you for inviting me to speak today. And so I have actually a very short outline for us today. And so I'll focus on the brain responses to interoceptive signals as measured by functional MRI, and then I'll discuss brain responses to intrinsic vagus stimulation, and finally how we can modulate interoceptive pathways via the vagus nerve. And along the way, I'll also point out some conflicting evidence and gaps in the field that could use some attention to help push us forward. So Ellen just covered very nicely the ascending pathway, which I was going to go through, but very briefly again, just to give you a bird's eye view before I jump into things. Visceral afferents are conveyed via autonomic nerves that travel with spinal nerves and synapse onto lamina 1 of the spinal cord. And as I mentioned, and as Helen mentioned as well, they also travel outside the spinal cord via the vagus nerve. And the sensory neurons of the vagus nerve are located within the nodose and the superior jugular ganglion. And these fibers project directly to the NTS, specifically within the medulla region of the brainstem. And the interoceptive afferent fibers from the spinal cord also project onto regions of the brainstem. And from here, they have widespread cortical and subcortical inputs that I'll get into now. Common way to probe interoception with fMRI is to ask participants to pay attention to body signals. And this could be somewhat confounding, but interoception inherently requires attention to bodily states. So studies sometimes use a variation of the heartbeat counting task and, the breathing detection task. And these signals are typically compared to exteroceptive stimuli, like as controls, like detecting a dot on the screen, for example, or looking at text that is blinking on the screen. And other interoceptive tasks used to probe visceral afferents include things like esophageal and rectal distension as well. And even though pain sensations can be somatic, it does actually fall under interoception. And there is extensive overlap between these pathways. But for this part of the talk, I'll focus on awareness of body sensations like heartbeats and breathing. So this particular study was interesting because they probed interoception that comes from a visceral source, and in this case, it's heartbeat detection, and also a somatic source, which was skin temperature detection. And they found that focusing on heartbeat versus skin temperature produced greater activation within the insula and sensory motor and limbic regions that we see here in yellow. And focusing on the skin temperature activated these regions, but to a lesser extent. And greater widespread activations seen in blue here are found within the primary sensory cortex and even the occipital cortex, despite the investigators using similar cues for each condition. So in this conjunction analysis here, we see shared regions that respond to focusing on both interoceptive conditions combined and both mapped onto the insular and sensory motor regions here in red. So we see that interoception from both sources share overlapping regions, but also have distinct representations as well. And in this particular study, the participants also completed the MAIA, the multidimensional assessment of interoception awareness, and it would have been nice for this talk to come after Wolf's, who would speak more about this. But they found the trend in a trending negative correlation where subjects with fewer heartbeat counting errors had higher scores on the first factor of their factor analysis. And this included characteristics like increased tendency to notice body sensations, for example. Now, this next study had very similar findings using a breathing task, and they also found that the more accurate a participant was at breathing detection, that the greater activity they had within the insula, and the investigators also found greater connectivity within the anterior insula to somatosensory regions during the interoceptive condition compared to the exteroceptive condition. And an interesting part of the study was that they also included a cohort of patients that had focal lesions that overlapped in the anterior insula, shown here in red. And so compared to healthy controls, and also compared to another set of patients that had brain damage to regions other than the insula and the somatosensory related regions, the insular lesion patients had decreased interoceptive, the whole panel is kind of in my slide here. There we go. The insular lesion patients had less interoceptive accuracy compared to exteroceptive accuracy, which was unaffected here. So this really suggests that the anterior insula plays a significant role in breathing detection. And this is interesting because a previous study, a case study by Kalsa, showed that heartbeat counting accuracy was only delayed and not really affected in a patient with insular damage. And the two tasks in this case are similar in terms of interoceptive focus, so it's really unclear whether the differences are methodological or neurological in this case. All right, so while it seems that I've been focusing mainly on the insula, remember that these studies also found activation within regions like the sensory cortex, the anterior cingulate, the temporal and frontal regions, and a vast literature has shown engagement of these regions during emotional and cognitive tasks as well. So in this particular study, they had 3 separate meta-analyses performed on studies about interoception, social cognition, and emotion. And through a conjunction analysis of these meta-analyses, the 3 meta-analyses, they found overlap between the insula, the ACC, and the amygdala and the hippocampus, which make up this insular frontotemporal network. And this really lends support to the notion that interoceptive signals converge and integrate with these other networks. And in the same paper, the authors compared healthy controls to patients with lesions within this particular network. And as we see here, the patients performed significantly worse in each of the interoceptive, in the emotional, and cognitive domains. So to briefly summarize so far, the insula plays a key role in interoception, and the insular frontal temporal network seems to be a convergent center. So we discussed the responses to paying attention to bodily signals conveyed via the vagus nerve, and so now I'll move on to brain responses that we've observed when we stimulate vagal pathways here with electrical stimulation. Now, the invasive approach to vagus stimulation requires an implanted electrode, and this electrode and the leads are attached to the vagus nerve itself, the cervical vagus nerve in the neck, and provides direct electrical stimulation. And this is typically used for patients with refractory epilepsy or depression. And because of the pathological conditions, it's been difficult to discern the extent to which the afferent vagal activity influences networks outside of epilepsy and depression. But to date, the mechanisms of action underlying these therapeutic benefits for these particular conditions are still not entirely understood. However, we now do have new non-invasive tools that allow us to gain access to the vagal afferent fibers via electrical stimulation of the neck and via stimulation of the auricular branch of the vagus nerve as well. So recently, using fMRI showed that we can access the primary vagal afferents from electrical stimulation of the surface of the neck just above the region of the vagus nerve, and 2 minutes of the stimulation compared to stimulation of the control, which was the sternocleidomastoid muscle, produced activation within the NTS and the medulla. And prior to this, I extended the findings of Dietrich and Krauss by showing localized NTS activation in response to electrical stimulation of the cymboconcha of the ear compared to stimulation of the earlobe. And the fibers of the auricular branch of the vagus nerve actually come from the superior jugular ganglion, as opposed to the nodose ganglion, which contains the sensory fibers that convey visceral afferents. But as we can see, afferents from both ganglia converge on the NTS. And most recently, the Napido lab meticulously mapped the projections of respiratory gated TVNS within the medulla using a 7 Tesla scanner, which produced these very nice high resolution images. And we could see here activity within the NTS. And this was also a part of a larger dose response like study. And prior to that, Yakunina and colleagues reported that optimal NTS activation was produced by stimulation of the cymboconcha, which is here, as opposed to stimulation of the ear canal or the tragus, which is right here. And we could see the differences here within the lower medulla. So using both approaches, I found that not only can we access the NTS, but we can also access vagal cortical and subcortical projections. And these approaches activated the interoceptive regions just discussed, like the insula and anterior cingulate, the primary sensory cortex, and the prefrontal cortex as well. So we've gone over so far the afferent projections of interoception and extrinsic vagus stimulation. So now I'll talk a little bit more about the effects of modulating these pathways with electrical stimulation of the vagus nerve. And Gina will actually be presenting very cool and novel methods to probe the regulating descending autonomic pathways. So I'll discuss modulation of more cognitive and affective and somatic sensations. So there is plenty of evidence that suggests that vagal stimulation modulates affect or mood, and so much so that it's an FDA-approved treatment option for refractory depression, and this is the invasive procedure. There's also evidence that it's beneficial for other treatment-resistant disorders like OCD or panic disorder or PTSD. And because because such a invasive nature, and because of the underlying conditions, there's actually been a very limited number of studies that specifically looked at the effects of invasive stimulation on cognitive function and pain. But we do see that improvement in things like logic, reasoning, and working memory, and some decrease in pain sensations has been reported. Now, it is a bit easier to study the effects of vagus stimulation using non-invasive approaches, but keep in mind that this is not necessarily identical to direct stimulation of the cervical vagus nerve. And there are several variables to consider when using this approach, like location and duration of the stimulation and the stimulation parameters, just to name a couple. But nevertheless, several reports now show that beneficial effects several reports show that the beneficial effects against depression, anxiety, mood, and well-being in healthy participants and patients with chronic pain or major depressive disorders using this non-invasive approach. And other studies have found that it improves memory, it improves learning and reaction time, and it reduces headaches and some pain sensations. But overall, the results have been mixed. And within these particular studies and studies not shown here, investigators have also found either no effects or non-responders. So the literature is still conflicting. It is suspected that the modulatory effects of vagus stimulation occurs by accessing the NTS projections within the brainstem. And these projections include regions like the parabrachial nucleus, the locus coeruleus, the Raphe nuclei, and the periaqueductal gray. And all of these regions were activated by both the non-invasive and by both non-invasive procedures. But it's really important to understand that that observed activation in a particular region doesn't necessarily imply release of these associated neurotransmitters. It really remains unclear what vagal-induced activation, for example, of the locus coeruleus means unless we actually measure levels of norepinephrine or find reliable proxies, which we have yet to do effectively or efficiently. And the same could be said about the activations within the cortex and the subcortical regions as well. And luckily, this is actually a very recent review that was just published, and it essentially came to the conclusion that the lack of the consensus among TVNS studies is in part due to methodological differences, and in some cases, the studies don't fully report their methods, which makes replication very difficult. But luckily, Julian Koenig really rallied and drafted a recommendation for minimum reporting standards, and specifically for TVNS research. And so hopefully this will be out very soon. So with that said, I'll continue and also start to wrap things up here. Now, because I've been concentrating on pain research, I'm going to shift my focus a little bit into pain, but this still falls under interoception. And so from studies outside of vagus stimulation, we know that emotion and cognition modulate pain differentially. So for example, positive moods decrease pain and negative moods have been shown to increase pain. And manipulating cognitive processes like attention can also modulate pain. So for example, taking someone's attention away from a painful stimulus will decrease pain perception. And these psychological factors influence pain perception differentially, such that manipulating emotion affects pain unpleasantness, while manipulating attention affects pain intensity. And now, because interoceptive afference or vagal afference accesses these networks, it's possible that vagal stimulation could modulate pain perception differentially. In fact, an early anecdote published by Brockhart from a patient with the implanted vagus stimulator to treat depression reported that his chronic low back pain no longer, quote unquote, bothered him after implantation. So it's unclear if the intensity of his back pain changed, but evidently the negative affective component of the pain was modulated by the vagal input, although I certainly don't dismiss placebo effects in this case either. Now, in this review paper, I also show that most studies examining the effects of TVNS on pain perception typically report only pain intensity ratings and not pain unpleasantness ratings. So it's unclear whether vagal afference preferentially modulates one pathway versus the other, or both equally. And knowing this information could help us design more specific interventions going forward. And of course, to this end, I have an ongoing randomized double-blind crossover study here at NCCIH, and we're about halfway through our data collection. But just to close up here, to summarize, afferent interoceptive signals converge at cortical and subcortical levels with emotion and cognitive networks. And we now have new and exciting non-invasive tools to probe interoceptive networks, but these tools and their methodology are still in progress. And while we can access and modulate these interoceptive networks, we have to remember that we'll also target multiple regions or multiple networks, and we don't have the methodological specificity yet to do so differentially. So it's important to consider adding measures that capture changes produced by these different overlapping networks so that we can learn more about how they're modulating vagal afference. And it's also evident that more work is needed to fully understand interoceptive afference and the subsequent cascade of regulating signals. And understanding these mechanisms can help us better understand the therapeutic effects of vagus stimulation and help design better interventions. And I really think that TVNS paired with multimodal approaches can help. And pairing tools like transcranial magnetic stimulation or magnetic resonance spectroscopy with fMRI could provide a deeper functional insight about how TVNS affects the efferent or the regulatory pathways of the interoceptive system, which ultimately could maybe one day have beneficial widespread effects on the mind and the body. All right, thanks to everyone, especially the Interoceptive webinar staff and the speakers for their feedback. So I will stop sharing now. And up next, I believe, is Jeannie.
Thank you, Eleni. That was great. So I'm going to now switch gears, and we heard a great presentation on the vagus nerve, and switch now to talk about micro-neurography as a technique to measure efferent sympathetic nerve activity and its potential applications in interoceptive research. So the sympathetic nervous system is the arm of the autonomic nervous system that we classically think of as responsible for the fight or flight response. So the cell bodies of the sympathetic nerves reside in the thoracolumbar region of the spinal cord and connect to sympathetic control centers in the brainstem. And then these presynaptic neurons then synapse with postsynaptic neurons in the sympathetic chain ganglia, and then go on to innervate various organs and tissues throughout the body. So the end result of sympathetic nerve transmission is a release of norepinephrine and other neurotransmitters at the nerve terminals that then will increase heart rate and cardiac output, increase sodium and water reabsorption from the kidney, and constrict blood vessels to increase blood pressure. So the sympathetic nervous system also, in addition to the fight or flight response, plays a critical role in blood pressure regulation, which is controlled by afferent input from the arterial and cardiopulmonary baroreflex systems. So the arterial baroreceptors are mechanoreceptors that reside in the carotid sinus, the carotid bifurcation, and the aortic arch, and modulate sympathetic activity in response to changes in blood pressure. So if the blood pressure goes up, then these arterial baroreceptors become activated, leading to a reflex decrease in sympathetic activity. And conversely, when blood pressure goes down, then the arterial baroreceptors become unloaded and lead to an increase in sympathetic activity. So this is all in an effort to keep blood pressure within a narrow range. So the volume-sensitive cardiopulmonary baroreceptors, on the other hand, are located in the atria and in the lungs, and they modulate sympathetic output based on changes in blood volume or venous return to the heart, as might happen with volume depletion or with upright posture, again, to maintain blood pressure homeostasis. So the sympathetic nervous system, again, plays a major role in both short-term and long-term blood pressure regulation. And there are a number of diseases, in fact, that are characterized by chronic overactivation of the sympathetic nervous system, such as hypertension, heart failure, chronic kidney disease, and post-traumatic stress disorder, that contributes to increased cardiovascular disease risk. So chronic overactivation of the sympathetic nervous system contributes to increased cardiovascular risk by increasing blood pressure, but there are also a number of deleterious end organ effects that are independent of blood pressure, by which sympathetic overactivation contributes to increased risk. So in order for us to be able to study the sympathetic nervous system in humans, we need to have a method, a reliable method for measuring sympathetic activity. And there are some here that we can consider. So norepinephrine is the major neurotransmitter that is released at sympathetic nerve terminals. But plasma levels of norepinephrine are not just the product of sympathetic nerve activation, but is also influenced by metabolism, reuptake, and clearance, and so is not necessarily the most reliable measure. While there are some heart rate variability measures that are valid markers of cardiac parasympathetic activity, the measures of heart rate variability that are purported to reflect sympathetic activity are actually less valid and less reliable. There are some studies that have shown that the galvanic skin response correlates with skin sympathetic activity, but not all studies have shown this correlation. The norepinephrine spillover technique involves the infusion of radiolabeled norepinephrine, and the major advantage of this technique is the ability to quantify organ or tissue-specific sympathetic activity. But the use of this technique is actually quite limited because of its invasive nature, and it's also a pretty cumbersome technique. And so, and also it doesn't allow for detecting continuous changes in sympathetic activity. So that leaves us with micro-neurography, which was developed 50 years ago by Valbo and Hagbarth, and is the gold standard method by which we can measure and quantify sympathetic activity directly in real time from awake humans. And so in microneurography, we place a recording microelectrode inside one of the accessible peripheral nerves in the body. In our lab, we use the perineal nerve, but other nerves such as the median nerve or posterior tibial nerves have also been used. And we can measure sympathetic nerve traffic in real time from this recording electrode. So to remind you where we are recording from in terms of interoceptive neural pathways, so in microneurography, we are measuring directly from these postganglionic sympathetic nerve fibers as they travel within the peripheral nerve. So in microneurography, we place a recording electrode inside the perineal nerve, and a reference electrode is then placed in close proximity. And when there is activation of the sympathetic nerves, this creates a voltage difference between the recording electrode and the reference electrode. And this multi-fiber signal is then amplified, integrated and filtered, and then displayed as a neurogram. So here is an example of muscle sympathetic nerve activity, or sympathetic activity that is directed to the blood vessels that supply the muscles of the lower extremity, and along with the concomitant EKG and concomitant beat-to-beat arterial blood pressure. And we can quantify muscle sympathetic nerve activity as burst frequency, which is simply the number of bursts per minute, or burst incidence, which is the number of bursts per 100 heartbeats, or total activity, which also takes into account the amplitude of these bursts. So here's a picture from our lab showing a microelectrode inside the perineal nerve. And this is a reference electrode in close proximity. And this diagram shows the microelectrode inside the nerve fascicle with the tip of the electrode next to these sympathetic nerve fibers. So here's an example of a neurogram from a patient with end-stage kidney disease. And you can see that muscle sympathetic nerve activity is substantially higher in this patient compared to the age-matched control. And these are the cumulative data that show that mean resting muscle sympathetic nerve activity is elevated in kidney disease compared to controls. And here are just some examples from another patient population, sleep apnea, showing elevated sympathetic nerve activity in sleep apnea compared to obese individuals without sleep apnea and normal weight individuals. So not only can we use microneurography to characterize baseline or resting differences between patient groups, but we can also study the reactivity of the sympathetic nervous system to stress. So in this example, we showed that the change in muscle sympathetic nerve activity is augmented in patients with post-traumatic stress disorder compared to veterans without PTSD. And this is important because exaggerated sympathetic reactivity is associated with an increased risk of hypertension and cardiovascular disease. So there are multiple potential applications of microneurography in interoception science, such as in the study of the regulation of the sympathetic nervous system by interoceptors such as baroreceptors and chemoreceptors in both health and disease. And also in understanding the effect of acute and long-term interventions that manipulate or target interoceptive pathways on sympathetic activity, reactivity, and regulation. So as an example, the classic method for quantifying arterial baroreflex sensitivity, and again, these are the stretch receptors that modulate sympathetic activity in response to changes in blood pressure, is through the modified Oxford technique, in which we give an intravenous bolus of nitroprusside to pharmacologically manipulate the blood pressure, which leads to a decrease in blood pressure and a reflex increase in muscle sympathetic nerve activity, with a reflex increase in heart rate and a reduction in the RR interval. And then this is followed by giving a bolus of phenylephrine, in which there's an increase in blood pressure that leads to a reflex dampening of sympathetic activity and a reduction in heart rate, again, all mediated by the arterial baroreceptors. And we can quantify the sensitivity of the sympathetic arm of the baroreflex by examining the relationship between these changes in induced blood pressure and sympathetic nerve activity, such that the steeper the slope of this relationship, the higher the sensitivity, and the flatter the slope, the lower the sensitivity. And so here in this figure, we showed that in patients with post-traumatic stress disorder, the slope of the relationship between diastolic blood pressure changes and muscle sympathetic nerve activity is significantly flatter compared to controls without PTSD, demonstrating an impairment in arterial baroreflex sensitivity. And this is clinically important because impaired baroreflex sensitivity is associated with an increased risk of hypertension and cardiovascular disease. So microneurography can also be used to study the effects of interventions that target interoceptive pathways to modulate sympathetic activity. And in particular, we have been interested in non-pharmacologic or mind-body interventions that may activate baroreceptor or vagal afferents via slow breathing, mindfulness meditation with a focus on breathing awareness, and vagus nerve stimulation. So in these studies, we showed that slow breathing at sub-physiologic rates of 5 to 6 breaths per minute using a biofeedback device acutely lowers muscle sympathetic nerve activity with a concomitant reduction in blood pressure in veterans with post-traumatic stress disorder when compared to a sham breathing device that directed breathing at normal respiratory rates. And also device-guided slow breathing, as opposed to the sham, improved sympathetic baroreflex sensitivity in PTSD. And in another study, using mindfulness meditation with a focus on increasing breathing awareness or breathing awareness meditation, we found that there was a significant reduction in muscle sympathetic nerve activity and mean arterial pressure and heart rate compared to the control intervention. And this was in a population of older African-American men with chronic kidney disease who had high sympathetic activity at rest and had increased cardiovascular disease risk. And so finally, there are some animal and human studies that suggest that electrical stimulation of the vagus nerve inhibits sympathetic activity. And these are data from the only study that has looked at changes in sympathetic activity using microneurography during vagus nerve stimulation. And they found that stimulation of the auricular branch of the vagus nerve leads to an acute reduction in sympathetic nerve activity. So the purported mechanisms by which these interventions target interoceptive pathways are that with vagus stimulation, there's activation of the vagus afferent nerve that feeds into the nucleus tractus solitarius, that activates the caudal ventrolateral medulla, which then inhibits the rostral ventrolateral medulla, which is the sympathetic control center in the brainstem, to reduce sympathetic activity. And with breathing interventions, including slow breathing and mindfulness meditation with an increase in breathing awareness, the reduction in respiratory rate leads to an increase in tidal volume, and this is to maintain the same minute ventilation. And so when that happens, there's activation of the pulmonary baroreceptors, which through the vagal afferents then inhibits sympathetic activity. And there's also a conscious component, a central feed-forward component as well, from an increase in breathing awareness that also has a sympatho-inhibitory effect. So in just the last minute, I wanted to introduce this methods paper that was just published last month in the Journal of Physiology. from Dr. Von Macefield's lab, in which he reports the first case series of successful vagus nerve recordings using microneurography. So these are really the first reports that show that the vagus nerve can be directly accessed and recorded from in awake humans. And it really opens up a lot of new exciting opportunities to study both afferent and efferent vagus nerve activity directly. So here's a picture from that report that shows the microelectrode going into the back of the neck under ultrasound guidance. And here is an ultrasound image that shows the tungsten microelectrode entering the vagus nerve here between the internal jugular vein and the carotid artery. So to summarize, sympathetic microneurography allows for direct intraneural measurements of efferent postganglionic sympathetic nerve traffic in humans in real time. And sympathetic microneurography can be utilized in studies examining potential benefits of interoceptive manipulation on sympathetic activation. And vagal nerve microneurography is a very new technique that offers new opportunities to examine both afferent and efferent vagus nerve activity directly in humans. So I will end there and turn it over to Wolf.
Okay, can you hear me?
Yes, we can. Wonderful.
Okay, so I'm gonna show myself briefly, but I'm gonna turn my video off to make sure that we have a good reception here and this works good enough. So I apologize, I'm on vacation and at the place here, the internet connection is not perfect. So I take my video off and then I'm gonna share my screen. Share screen here. And then I'm gonna get this over here and go here. And how do I get this full? Can you see my screens now, my slides?
Yes, we can.
Okay, perfect. So I have to find how to move this. Yeah, okay, here we are. Okay, so I'm talking about mostly conscious aspects of interoception. So we heard this fantastic physiology data that are very, very encouraging and very good. So my background is that I'm coming from studying mind-body interventions and talk a lot with practitioners of these interventions. And I'm trying to understand what's happening within interoceptive awareness in the area, what we can perceive actually. I have to see this. Is this going on here? Okay. Is it better? Yeah, I can think. Okay. So I'm talking about the aspects that are accessible to conscious awareness. And first, I want to bring some data that I got from the NCCRH-funded qualitative study, where we had a group of mind-body interventions, practitioners and patients together or in separate focus groups, and they talked about what they understand, how they relate to, how they understand the relationship of mind and body. So they actually all insisted first to make a statement that mind and body are not viewed as separate entities. There's an integrity of the self. And second, that there's a human capacity that's innate to all of us for a developmental process to embodiment, to further embodiment. So that's a statement that these mind-body practitioners made. I'm going to come back to. So the environment in which this mind-body intervention happens within the Western culture is characterized to some degree of disembodied quality. Our global civilization has an increased dependency on the internet and social media, and both are shaping self-identification, self-objectification, and how we perceive our body. And we're spending increasing hours in virtual reality, doing Zoom conferences and going to cyberspace. And these are all challenges for the integrity of mind and body. If some of you guys might have read Harari, who's one of the best-selling authors these days, philosopher and dedicated meditator from Israel, who in his last book, 21st Lessons for the 21st Century, one of his most quoted sentences is, if you don't feel at home in your body, you will never feel at home in the world. So this developmental model of body awareness shows kind of 4 different levels that we encounter actually within our studies. So the first level is called the absent body, is when people take the body for granted. This is when they're young and don't have ever pain, teenagers or healthy volunteers. And then when they get into pain or some dysfunction in their system, when they get disrupting symptoms, then they find that these symptoms are kind of opposed to them, and they perceive the body as kind of an enemy they have to fight against, because that changes their possibilities for activities. Then another level is when they get into yoga and mindfulness, that they develop a new relationship to their body, which is characterized by cultivating the immediacy of perceptions and accepting the body. So that's the people who are the novices or graduates from mind-body interventions like mindfulness and yoga. And then the fourth level is when they are more experienced and when they're practitioners, then they get kind of the sense, the unity of body and mind, and they learn that the body can be actually a source of learning and meaning, and maybe they overcome the duality of their perception. So this is from a study by Kathy Kerr. She died a few years ago. She was at the Ocean Center at Harvard and then at Brown. And she titled one of her big papers, Mindfulness Starts with the Body. And she found that the that MBSR interventions, MBSR, MBCT, mindfulness interventions, change or modulate the alpha rhythm and in the brain. And this alpha rhythm in the brain is actually a filtering station at the level of the thalamus for how much sensations of the body are let through to the somatosensory cortex. So she did kind of one of the first computational models of how mindfulness or mindfulness-based interventions actually change brain rhythms and brain activities. So the developmental model also we discussed together with the predictive coding model, which is one of the currently leading theories on how interoception psychologically works. Predictive coding model means that when we have a perception of our body, for example, of ourselves, it's always a mix of predictions about our body, like based on our prior experiences, all the stories we're telling each other about ourselves on one hand, and afferents of real, actual, momentary perceptions on the other hand. So these all get mixed up. And for example, in the first stage of interoceptive awareness or body awareness, these are all mixed up with each other. So thinking and sensing are pretty much one mix. And in that mix, we can give different weight to what we perceive from the body itself compared to our predictions. So in short term, you can say between thinking about our body and sensing our body, what weight we give to each other influences the resulting perception of ourselves. And mental health problems, also according to the Kalsa lab, which does a lot of interoception research, is the dysfunctional weighing of these mechanisms, which governs the relative influence of prior expectations versus afferent body signals in determining our resulting perception. So that's a predictive coding model, which is kind of one of the leading theories now in neuroscience. So what I could speculate, but nobody has done this systematically, that also these 4 levels might relate to different brain function states, like the narrative mode, thinking about the body and playing with the body in a naive person is similar to the default mode network. Then we have chronic pain. Then in this balance between expectations and real perception, it's all getting dominated by emotions. On the 3rd level, we can finally, that FARP has shown that with a mindfulness study, we can finally differentiate between thinking and sensing about the body as 2 different paths, neurologically separate for self-identification or self-reference. And on the 4th level, then it shows that if we have more interoceptive attention, that actually the cortical areas in the brain can change. So I'm talking mostly about the regulation, which is on the right side here of this slide, more than anything else, and how this all works out in terms of regulating the system. How does it happen? So now I'm presenting some experiential findings, and the physiological details, I don't have much of those, so I'm glad that we saw those already by the previous presentations. So NCCIH supported a team that I led to develop a probing instrument, kind of a self-report measure called the MAIA, Multidimensional Assessment of Interoceptive Awareness. It has been translated now in many languages. And this is an instrument with 8 different scales, all different aspects of interoceptive awareness that are kind of correlated to the different stages of the developmental model. One of the key scales within this questionnaire is the scale for self-regulation, which stands for the ability to regulate the stress by attention to bodily sensations. So it's similar to what we had heard before, like related to breath awareness. So the goal of self-regulation is to create homeostasis, that we don't have pain, that we don't suffer, and a result when the prediction error, the tension between real perception and prediction or mental thinking, when this goes to 0, so that we can learn, for example, with mindfulness intervention, that they have 2 effects. One is they update the expectations, and the second is they shift the weight more to the afferent perceptions in the real time right now, present moment, rather than what we think about it. Now, this goal for self-regulation might be different in other cultures. There's a paper we just got into press where we studied Japanese Americans, which have a different goal. So the important thing, what I want to emphasize is here that the MAIA scales assess regulatory aspects of interoceptive awareness, and they differentiate between adaptive and maladaptive attention styles. So you can either have a mindful, present moment, curious, even-handed, kind of neutral, accepting attention style towards, for example, pain or other discomfort sensations, or you can have a hyper-vigilant, anxiety-driven, maladaptive attention style. And this has to be differentiated when you talk about interoception, because interoception can be maladaptive if you feel too much from your anxiety-related sensations. That doesn't help you very much. So this differentiation is not happening when you do the heartbeat counting test or heartbeat detection test or other accuracy measures. That's why I believe we need this differentiation in addition. So I bring 7 examples of self-regulation where particularly use the MAIA scales in most of these studies, where they showed that self-regulation is an important element for mind-body, that changing its self-regulation are the important factors in mind-body interventions. This is from one of the largest studies ever on meditation in Berlin at Leipzig, Max Planck Institute, where they found that self-regulation here was the largest effect size compared to controls, attention regulation the second, and so all these regulatory aspects of interoceptive awareness were the key elements in my body interventions, and also shows that different aspects of interoception change in very different degrees. And this is from a brief intervention study, mindfulness MBCT cognitive therapy for depression, which shows a sequential mediation. The treatment itself, which is like 4.5 hours of mindfulness related to depression, changed regulatory aspects of interoceptive awareness assessed by the Maya. That in turn changed decentering or de-identification from the thoughts about your depression. And that in turn mediated the changes in post-intervention depression. So you have a sequential mediation where the interoceptive awareness is one element of the process that happens in the mindfulness treatment for depression. This is another sequential model of mediation where here done at UCSF, where we did a study on mindfulness-based childbirth and parenting in women who were pregnant with control. There was a control intervention, and we found that the intervention, mindfulness-based childbirth and parenting before birth, changed postpartum depression via, again, a sequential mediation mechanism by changing attention regulation, another regulatory aspect of interoception, which in turn changed diminished rumination, which is one of the key elements of pain catastrophizing, which then in turn changed depression postpartum and even had a trend toward lower opioid use while these women were in labor. Another study, this is Cynthia Price's work, Mindful Awareness in Body-Oriented Therapy. She uses the Maya scale and showed that they had improvements with this mindful intervention. Women with substance use disorder are undergoing treatment, changed all the main outcomes that they wanted to know. And this was without a mediation analysis, but in parallel to changes, again, in self-regulation, attention regulation, and in body listening within the interoceptive awareness process. Now, this study is from Peter Wayne's group at Harvard Osher Center, where they did a Qigong study. This is only a single arm study, but it's improved several outcome measures in post-surgical pain patients from breast cancer surgery. And again, that was in parallel to key regulatory outcomes of interoceptive awareness. Now, this is the study here from actually Catherine Bushnell's lab at the intramural NIH, where they found that cross-sectionally, yoga practitioners, long-term yoga practitioners, had a clearly improved pain tolerance for experimental pain, which went along associated with increases in insular cortex, which we already heard is one of the hubs for interoception. And this also was correlated with the years of yoga experience. So the longer they did yoga, the more changes they had in the insula. Now I'm showing this for this reason on the left side of this slide. You see that how they dealt with this experimental pain. You know, this was a cold pressure hand and ice holding test. So the people who did yoga, they accepted the pain, they relaxed, they observed what they perceived, and they kept breath awareness. Whereas the controls distracted themselves, ignored the pain, and didn't have and had a much lower pain threshold. So this is unfortunately only a cross-sectional study, so we should do that longitudinally. So the last slide I want to show here is a study by the Abkarian lab, who's doing a lot of NCCRH reviews on brain processes related to chronic pain. Vachon Prasseau, who is a Canadian now in Canada, published some study where they found that the Maya was actually dominating one of 4 neuro traits. So they try to distinguish different types of people that have chronic low back pain and try to phenotype them into different patterns of sub traits. So the Maya, actually, the regulation aspects, again, self-regulation and tension regulation, were the key elements of one trait. And these scales, together with emotional awareness, that how you aware how emotional states are related to body sensations, these 3 scales were the only scales out of 19 different questionnaires that they used that were able to predict treatment outcome. In this case, it was a placebo study for chronic low back pain. So all I wanted to say, we need to do physiological measures like what was presented already, but we also should use questionnaires because they're different. They're able to differentiate different dimensions of interoception and also the beneficial from the maladaptive type of interoceptive awareness. And I'm finishing this by mentioning my main co-contributors to this research in the past. So I take it off here. Sorry, but it takes so long.
Thank you, Dr. Malin. We will now go into the panel discussions. We'll have the IC presentation. So the first one is Dr. Jean Simon.
Good afternoon. Thanks for having me. So I just wanted to touch on the National Institute on Aging's approach to everything, including interoception. So in general, I think that any of the techniques and methodologies at behavioral, psychological, neurophysiological levels are going to be relevant to the National Institute on Aging because the scope of our institute's goals is very wide. We're trying to understand the basic biology, understanding changes that accompany aging as distinct from that underlying specific diseases, recognizing also that people with aging, of course, develop a multitude of diseases. That includes understanding sensory and motor changes, which includes interoception, and including how neurobiological and psychological and behavioral interactions change and affect each other through age. Any of the approaches that have been described today could be relevant. The most important thing I think to emphasize here is that asking investigators when they're thinking about interoception to take a lifespan perspective as well and consider including longitudinal studies and recognize that interoceptive sensitivity measures or self-reports of interoception may change with age or with cognitive change. I'll leave it there.
Okay, thank you, Janine. I'm Jim Knott from NINDS, and I will just quickly use this slide to show how NINDS's interest in neurology, disorders of neurology, overlap with the symptoms and the disorders with interoception. And I would really just call your attention to the rightmost column there. As also being part of the Brain Initiative, we've come to realize the real power of research that can develop and help apply approaches that have a cellular resolution to understanding the system, that gives us a mechanistic understanding at a cellular level and a level of dynamic circuits and pathways through the brain. So we have particular priorities for that. So thanks to Vin and the speakers today.
Hi, I'm Stephen Grant from the National Institute on Drug Abuse. And interoception is a key issue in understanding the actions of drugs of abuse. As was outlined in the talks today, there are receptors for all of the major drugs of abuse on visceral end organs, peripheral interoceptive motoric pathways that were described, the central pathways that were described, and self-reported interoception is a big part of what people report when they take drugs of abuse, and even forms the basis of animal studies such as drug discrimination. In substance abuse disorder patients, dysregulated responses to cortical interoceptive brain networks, particularly the anterior cingulate and the insula that were emphasized in the first talk, are disrupted in patients with substance use disorder, as is subjective interoception, both sensory and metacognition, that is monitoring the concept of insight and its converse, denial, which is a key concept in substance abuse treatment. But the contribution for interoceptive processes and their alteration by drugs of abuse, how that contributes to substance abuse disorder remains unclear, and therefore is a big gap in our knowledge. So advances in this area would be very much related to the mission of NIDA.
Hi, I'm Tia Vaziri from the National Institute of Mental Health. So I'm just going to briefly outline a few points here that we feel are important to consider for interoception research in mental health, in studies looking at the role of interoception in psychopathology, or exploring at a more basic level how it impacts or is influenced by cognitive, reward, arousal, or regulatory systems. And so really the first point to consider is really the importance of having standardized behavioral tests for measuring different features of interoception, and some of these were referred to by the previous speakers. To the extent possible, these tests should have psychometric properties that allow for a quantitative assessment, and to be also compatible with methods that measure neural activity, such as fMRI, EEG, or potentially intracranial recording methods when clinically justified. The second point is really a consideration for causal experimental design to enhance mechanistic understanding and to ultimately really try to explain the cause and effect relations between various aspects of interoception and psychopathology. The next point highlights the importance of computational modeling, which helps provide a quantitative framework for understanding the complexities of interoceptive processing. And for example, one of these was mentioned by Dr. Meiling. There are computational theories that exist on how the brain estimates internal body state by combining afferent signaling with our prior expectations. And finally, on a more translational consideration, it would be clearly valuable to identify clinically sensitive interoceptive measures that could potentially serve as biomarkers for disease state.
Okay. Dana, would you like to speak for a minute or so?
So my name is Dana Schlosser, and I work at the Office for Behavioral and Social Sciences Research, and we are a coordinating office for the NIH. So we don't directly fund grants, but what we do is promote the behavioral and social sciences across the whole of NIH centers and institutes. As far as interoception, I think from the presentations that you've seen today, you can certainly see how behavior is intricately linked to interoception and interoception processing. And as I have on the slide, or just pointing out a few of the more behavioral leaning aspects of this, such as the self-regulation, emotional regulation. There's been research out about the role of interoception in social connection, as well as when things go wrong and you have issues with interoception dysfunction and mental health, where it's been implicated in mental health, as well as other disorders such as obesity. Another potential area of interest is also the differences that you would see between different cultures and social economic statuses between different populations and the differences in the interoceptive processing in these populations. And I will leave it at that. And thank you very much for all the speakers today.
Thank you, Dana. So I will speak very briefly from NCCH perspective. This is Wen Chen again. As you've heard, actually, many of the talks today, almost all of them have talked about the interactions between a lot of the complementary integrative health approaches include predominantly in the mind and body practices domain, and how they may modulate the various steps of the interoception processes, as well as they might even be able to serve as a tool to help us probe those processes. I also wanted to take this opportunity to point out that the other half of our portfolio really includes natural products, which include botanicals, probiotics, microbial, the gut microbes, for instance, as well as special diets, dietary supplements. And many of those might also be able to modulate and the interoceptive process, in particular the gut-brain axis. I just wanted to bring that into the picture as well as how important they may be, in addition to all the very important aspects that we touched upon today by the speakers. I would basically leave at that, and maybe we'll open. We are running a little short of time. We have about 6 minutes left for question and answers. We've received quite a few questions. I would like to have all the speakers and the panelists turn on their video, and so that we can answer questions. We will probably not be able to go through all the questions people submitted online. And if you haven't done it, you can, if you watch through the NH video cast, you can scroll down on that webpage and you will see live feedback, and there's a live feedback form. You can type in your questions. Please send in the questions, even if we cannot answer them today on the video cast. We will try to get back to you by email with the answers. So now, first of all, we will, the first question I would like to pose to the speakers and panelists, I think it's primarily directed to Dr. Langervan, regarding the definition of interoception, which we, as Dr. Langervan mentioned, include concept of regulation. So the question is, does it address generation of the internal signal? Alain, would you like to take it?
Sure. Yeah. The answer is clearly yes. Yes. That's part of the definition of interoception as we talked about it today. And so there might be other ways to define interoception, but this is how we define it for our purpose of this framework.
Yeah, I'd like to add in as well that the regulation part of the interoception is primarily really meant to say regulating the generation of interoceptive signals. Certainly there are regulation of bodily functions that many times may overlap with the generation of interoceptive signals, but not always. And then we wanted to keep those distinctions in mind. Okay. Anybody else from the audience would like to chime in on that particular issue? All right. If not, we'll go on to the second question. The second question is related to maybe as opposed to Dr. Franco, it's about interoception and auricular acupuncture. So have you looked at the acupuncturist's way for vagus stimulation? The results sometimes not very consistent, but there are convincing evidence. And also, what are the sort of important research direction in the future to address those, to solidify the evidence of its potential importance and relevance? Would you like to take it, Lenny?
Yeah, sure. So I personally have not done any acupuncture research, but it's certainly a viable approach. And if I understand correctly, there are acupuncture procedures that do involve some form of electrical stimulation as well. And so if you actually look at the acupuncture map of the ear, the region that we typically stimulate, or that some studies have stimulated, which is the cymboconcha that I presented, if you look at the acupuncture map of the ear, what you see in that region is the viscera. And so it seems like this information has been known for several hundreds of years, and it's only now that we're beginning to rediscover this using different types of tools. So it's certainly a viable alternative. I agree that there have been mixed findings, but I personally have not done this type of work, and I hope this addresses the question.
Okay, and also I wanted to make sure our panelists and speakers have a chance to ask questions to each other. I know, Eleni, you had a question for...
Sorry if I'm jumping. Hi.
Sure, yes.
Hi, this is Alexandra Vicentek from NIMH, and I just presented our little thinking about interoception, which is a very novel area. So I want to ask Eleni, I guess, have there been any studies that have actually looked at the neurobiological changes after non-invasive vagal stimulation? So I'm aware there are fMRI studies, for example, examining vagal stimulation in fear encoding, and what they have looked mostly is whether they're actually engaging some of the projection targets, like nucleus-solitary tract. But I'm just unaware of any studies that they actually looked. Okay, so whether this improvement in responses to fearful stimuli coincides with some of the known biological targets, like let's say the connectivity between the prefrontal brain to the limbic and so forth. So I was just wondering, I guess, question for you and for anyone on the panel.
Yeah, so the literature on transcutaneous vagus nerve stimulation has just really grown exponentially in the past 10 years, and information about connectivity analysis using fMRI approaches or other biological measures like pupil dilation measures or activity of the heart rate variability changes. That has been published, but again, even within these different types of efferent effects, it's really still unclear as to what are the specific parameters that should be used to induce these type of effects, and it's unclear what location to stimulate in, what is the appropriate control to use. And so these are still outstanding questions, and this is what we hope to really consolidate going forward with this consensus paper that is under review right now. So hopefully we can get better answers to these questions. But yeah, there is a lot of literature in terms of differences in connectivity analyses as well, particularly in the most that I've come across is within the realm of effects of TVNS on depression or anxiety. Yeah.
I know we are running out of a little time, but I just got a message saying that we can stay on for a few extra minutes, so I'll try to see if we can extend for another 5 minutes to answer some of the questions. I see questions are coming in. I will read the question. This is a question for the representative from NIDA. So Steve, this is for you. The question, the person, this is Mark Enderman from Harvard Medical School. His question is, I'm wondering if studies exist delivering analogs of the drug of abuse that's engineered not to cross the blood-brain barrier to see whether the peripheral actions drive interoceptive sensing of drug context that in turn triggers release. Steve, you need to unmute yourself, please.
I wrote a text reply to that response because I didn't know whether we would have time. The short answer is there have been studies, particularly of cocaine methyl iodide, which does not cross the blood-brain barrier. That drug is not self-administered. However, it still leaves open the issue of other drugs of abuse, how much peripheral actions may contribute to the self-administration, and the further issue of how much peripheral action, interoception, plays in the process of addiction as opposed to drug action. So it's still very much an open question, and we don't have a large armamentarium of pharmacological tools to test these hypotheses.
Okay, I hope, Mark, you're happy with the answer. And the next question is for Dr. Franco. It says, Dr. Franco mentioned the role of norepinephrine release upon vagal stimulation. The audience is wondering if there are any implications of this to autoimmune and inflammatory conditions and their treatments.
So there is evidence in both animals and humans that norepinephrine is released in response to vagal stimulation. As to whether there is a direct relationship between norepinephrine and autoimmune functions is unclear to me. It's very outside of my field, but if you're interested in that line of work, then Kevin Tracy's work is really where you want to look at, and I think you may find your answers there. I would venture to say that yes, there is a role for vagal stimulation induced release of norepinephrine on inflammatory conditions based on Kevin Tracy's work.
Definitely, the interoceptive circuits are very intertwined. One process could, as shown by, I think the talk from Jenny Park, that even if you modulate the vagal process, it could be actually regulating the sympathetic activity and vice versa. So I think there's a lot of opportunity, research opportunities that exist for people to explore. There's another question from the audience. Would like any of the researchers and maybe the panelists as well to speak about whether they're aware of any research, current studies involving dance movement therapy and interoception. And they even mentioned a very specific method called the Feldenkrais method. I don't know if anybody would like to take a response for that. I mean, this is really, dance movement therapy is in many ways, I mean, it's a very complex therapy. Obviously, it has many components. Some of the components could be modulating interoceptive processes. I'm not aware of anybody looking at its modulation of the interoceptive process, although I wouldn't be surprised if it would have an effect.
So I actually gave an informal lecture to a dance group this time last year on this topic, and the scientific literature obviously is very thin, but when you think about proprioception, the interoception of your body in space, and particularly vestibular function and the circuitry in vestibular function, it converges on the same pathway, central pathways that were described in the talks today. So I wouldn't be surprised that there was a substantial effect. And in the context of substance abuse, I've thought about dance therapy and other types of movement therapies, including Tai Chi and martial arts, that it might be beneficial in terms of, not in terms of interoception per se, but in terms of our favorite system, the dopamine system and the striatal complex, that movement therapies could be a way of stimulating those circuits that get blunted after chronic drug abuse. So it's a rich field, it's a rich opportunity, but there is not been that much work that I am aware of.
Okay, great. Thank you, Steve. So I think, Eleni, I know you have one question for Dr. Ginny Park, and I think it's an important one. Would you like to post it directly?
Sure. So I was wondering whether using this micro-neurography approach, would we be able to differentiate the neural, I'm sorry, I'm getting feedbacks from somewhere. I don't know. Well, would we be able to differentiate the neural efferent and afferent activity with this microneurography approach in the cervical vagus trunk, which is actually composed of 80% of afferent fibers?
Yeah, so that would be a great question for Dr. Macefield, who was the, who did those first initial studies last month. But you're right, the vagus nerve is so heterogeneous that it's going to be essential to be able to determine what we're actually recording when we're recording from the vagus nerve. And so they were able to tell that they were recording from cardiac efferent nerves because the nerve signals co-varied with physiologic signals in the heart. And so in that way, they were able to determine, okay, these are cardio-inhibitory axons that we're recording from. Some of the afferents, though, you may not be able to readily measure the physiologic signals coming from the gut, for example, to know that you're measuring afferents from the gut. So I think, if there's some way that you can manipulate the physiologic signals and then that correlates with the vagus nerve impulses that you're recording from, that you can make that determination. But I think it's going to require, this is a big breakthrough in the field, but it's probably going to require more work to work out all those details.
Thank you, Ginny. I think we have questions for only one more, time for one more question, and this is for Wolf Malin. Will mindfulness interventions be expected to help people who find panic-inducing to tolerate physical experience and increase carbon dioxide from mask wearing? Obviously, that is very relevant to our current pandemic situation. Wolf, would you like to take it?
Well, I don't think there's any studies that have been done on that. I don't know. So, panic disorder is related to symptoms like heart beating faster, cold feet, all kinds of physical sensations that are related to panic. And if you wear a mask, you might feel some dyspnea, which is also an interoceptive provocation method. So theoretically, it's possible you can find something there. I don't know.
Okay, we are really running extra time, and I think we kind of have to close. I'll let Dr. Emmeline Edwards to close the event today.
Hi, everybody. I'm Emmeline Edwards. I'm the director of the Division of Extramural Research at NCCIH. And it's unfortunate that we have to come to a close for this wonderful session. But before we do, I want to thank the speakers and the panelists for their wonderful presentation and the discussions. I also want to thank the audience for their participation. Obviously, this is a very interesting topic, and as we heard, there's a lot of research gaps, and I look forward to having more development in this area. Before we close, I'd like to emphasize that we're going to have more frequent hot topic webinars at NCCIH. So keep an eye out on our website. I do believe the next one will be in September on the concept of implementation science. So keep an eye out. So thank you very much again. It was a wonderful session.
Okay, thank you, everybody. Have a great rest of the day, and thank the audience for joining us too. Bye-bye.
Bye-bye.
Thank you.